to whom it may concern. 50% of the email, but all email collected between this date and this date will be severely deleted.
I love that.
So if it's important, please read me when email me
Yeah.
this date. And I did. At the end, I got rid of 840 emails.
What a good idea.
Just one big delete.
Yeah. I like that. I mean,
Well, if you're going to take a vacation, so many times we take some time off, we'll still look at the email. It's like, well,
you see that?
I
I'm honorary and giving myself a degree now. There you go. It's a good day. It's a party.
I'm honorary prime minister.
Wow. I don't want your job.
I don't either. I already resigned.
So there won't be any people chanting your name. Fire Ed, Fire Ed. We have everything. I have everything. Okay. Yeah, Wendy is taking care of it.
She's all over.
Yeah, it's so nice to have somebody who knows how to focus on it. Because it adds exponential stress when you have to do it lunch and deal with like errors, and now it's just to show up and say, hey, how's it going? Yeah, it really makes sense.
The hour right there.
Yeah, that's right. Yes. I get to go to meetings. But. Hey, Sue.
Hey,
How are you?
let me know. I'm going to be back to anything.
Okay, next.
Hey.
Yeah. Just to update my Blazer ID, and now my phone's like, what? Can you get on there? Okay, yes. No, Sunday we went for a hike, which I thought we were going to go for a 3-hour hike. But I forgot that when there's snow on the ground, you don't have shoes. First of all, you can't see the trail. And every 10th step, you're like, you're a snow tire. So we were undeterred. We had a good nap. Had a good view. So we hiked for 6 hours. We were so tired. But yeah, at some point, at some point, no return. Like either we can go back and then we can for sure hike another, X hours, or we can get to the exit point where we think it is. We end up in this meadow, and you can see forever, which is really annoying because you're not making any progress visually. The barn. Hiking for 40 minutes. The barn's still far. It was, but my calf and everything, so. We used to do that. So we saw a coyote, which is not beneficial, but it's really pretty when it's like someone around. And moose.
My wife and I used to go out there all the time. Years ago. For whole childhood. When her and I got together, we went out there. We were close to her. We had a bar lazy after ranch.
Okay.
And,
Yeah.
Fabulous. We're out there on horseback, riding wherever we want to go. Buffalo,
Yeah.
of course, free farm.
But yeah, we almost got hit by buffalo in the car. The main herd was on the left, and this bull wanted to join his parent probably, but he charged in the road and then made a U-turn right before it hit us, so that was kind of good. But you realize, like, there is nothing you can do. If he decides that you're not his buddy, you have a problem. No, I'll just. And then yesterday when we flew out, they actually said that the last time of the season, usually the red doesn't work on the screen. No, the only way you really can With the mouse, it's kind of showing that with the whole new LCD system. I think it's some three doors. Go ahead and open the chip back. Come on in, guys. Good morning. I don't think it works on the. As the last folks are trickling in and getting some lunch, thank you all for coming to today's lecture in the Drug Discovery Lecture Series. Today we're going to talk about something very important. My thesis was in this topic, so of course it's the most important topic ever. Pharmacokinetics, and I think it's a subject that most of us are not that familiar with, or it's not on our radar screen typically when we think of drug discovery. What happens a lot is you discover a molecule that seems to work in cell culture and it kills cancer cells, or it cures diabetes, or it protects neurons from dying, and you think, wow, let's stick it in a mouse or stick it in a rat. And it's a seed that treats the disease, but what we forget is, well, do you know that you reach appropriate concentrations in the bloodstream or in the brain or in the pancreas or where it needs to go to have its effect? Also, what happens to how does the body deal with the drug? Does it chop it up? Does it stick it, excrete it in the urine? There's a lot of parameters that influence the behavior of drugs in the body. So I'm glad that we have Dr. Acosta here to talk to you about that. I kind of shifted topic. So pharmacokinetics is a little rusty in my brain, but he does it every day. And he's in the department almost every day, except Saturdays and Sundays. He's here in the UV Department of Pharmacology, Toxicology, in the Division Clinical Pharmacology. And he does a lot of the PK studies for example, the collaborative antiviral study group. So he knows a lot about antiviral drugs, but is also heading up the Pharmacokinetics core in the Cancer Center. So when it comes to PK on campus, Dr. Acosta is your man.
Because I'm the only person. Well, thank you very much for all of you for coming. Thanks, Michael, for that introduction. As she said, I'm the professor and director of the Division of Clinical Pharmacology in the Department of Pharm Tox and director of the Cancer Center's PK/PD core. I've been working in this field for, I don't want to say the number of years, more than 10 years. I'll just leave it at that. Way more than 10. And mostly I deal with antiviral drugs, antiretrovirals, and other antivirals, spanning from elderly patients afflicted with various infectious diseases all the way down to premature babies. And so, especially when you get down into the pediatrics group and the young babies, is really where the pharmacokinetics really come into play. A lot of what I'm going to talk about is more clinical pharmacokinetics, or pharmacokinetics that you normally see during the typical phases of drug development. But a great deal of this is applicable to animal studies in particular as well. We've done a lot. My lab doesn't do animal work, but we work with a lot of different people, either on campus or around the country, who do a lot of animal work. And so all of this is applicable there as well. I'm going to start off with a little bit of background information just on drugs, drug discovery, and some of the clinical pharmacology of the ADME, or absorption, distribution, metabolism, and excretion system that we think of when we have to think about pharmacokinetic data. But as many of you are, I'm sure, are aware, there's drug discovery can be a bit of a crapshoot. There can be up to 5,000 to 10,000 compounds screened, and over time, possibly one might get approved by the FDA. This can be a long, expensive process. I believe the typical cost of bringing a drug to market is now up to at least $1 billion or more for one compound, and multiple studies, obviously, along the way, phase 1 through 4, that need to be completed. If you've ever noticed, look at some drugs and the phase 3 develops. So phase 3 is the big clinical trials for whichever disease state. A lot of companies had to do multiple, like up to 4 or 5 different phase 3 clinical trials. A lot of times that might be because they're looking at a different patient population or different indication, but it's also sometimes because they don't have the right dose going into phase 3. Phase 2 is really where the dose needs to be found, the appropriate dose for that population. And so some of those items are what I'll be discussing today. Typical phase 1 type studies, and again, a lot of this is applicable to animal data, animals as well, but the single ascending dose and multiple ascending dose type study designs need to be done. The single dose, single ascending dose design is just what it says. It's only a single dose is administered, but a wide range of doses are administered, but only one each time. So it's a single ascending dose. One of the reasons for doing this is to clearly understand the kinetics of the drug. This might be mice, it might be people, it might be rats or monkeys. But when you go from, say, one of the drugs I've looked at recently, it was a 10 milligram single dose, all the way up to, I think there's 12 different doses, up to 1,600 milligrams as a single dose. One of the reasons you want to do that is to establish whether or not the drug has linear or nonlinear pharmacokinetics. And so the area in the curve, for example, produced by a dose of 20 milligrams would be doubled for a dose of 40 milligrams. That would be linear pharmacokinetics. If the dose is doubled from 20 to 40 and the AUC goes from 10 to 300, you've got a nonlinear drug. And that's something that you absolutely have to understand. Otherwise, when you try giving doses, you could, if you're in an animal model anyway, you could kill them without knowing why. Multiple ascending doses are similar, except that they're dosed to steady state, generally to steady state. So a wide range of doses are used, but multiple doses are administered to achieve steady state of the drug. And again, that's simply to look at if there's an accumulation of the drug. If the drug has a very long half-life, how much does it accumulate in the animal or person over time? Phase 1 studies also look at typically look at multiple formulations of the drug, food effect, food, no food, high-fat meal, low-fat meal, water versus a milkshake, things like that. Bioavailability studies and further assessments of ADME, the absorption, distribution, metabolism, and excretion of a drug. Phase 2, I generally think of much more as the actual dose-finding part of the clinical drug development process. This is where, and I'll touch on this and show a really, really good example of this towards the end. But this is where a wide range of doses need to be used, along with assessment of whatever the clinical biomarker for that disease state is, assuming that there is one. In the realm of HIV therapeutics, where I've been working for some time, we have the plasma HIV RNA, or the viral loads, that can be measured. And it's an excellent marker of response to drug therapy. And so this is where hopefully the appropriate dose would be found, and they're brought into the larger phase 3, typically 500 to 1,000 or 2,000 patient large-scale clinical trials lasting 48 weeks to show long-term safety and efficacy. And special populations, so dissecting out maybe older patients versus younger patients, looking at men versus women, issues like that would be looked at or sorted out from the phase 3 data. Phase 4 then is obviously going to be the after approval or post-marketing assessment of the drug. Many of the companies, once they get approval after phase 3, have to agree to phase 4 commitments. Typically, those are going to be drug-drug interaction studies that haven't been done yet, but need to be done. If it's a drug that could be used frequently, say, for example, in pregnant women, that population would need to be looked at as well. And also at the end of phase 3 and into phase 4 is where a company would make the decision, are we going to bring this drug into the pediatric population or not? So in HIV therapeutics and a lot of the antiviral drugs you deal with, there's a huge need to have those drugs in the pediatric population. And so those types of studies generally would get started, or at least developed, right around phase 3 or phase 4. So in clinical drug development, pharmacokinetic aspects of a drug can and have caused drug failure. These can be due to poor bioavailability of the drug for an orally administered compound, due to low solubility or high first-pass metabolism, inadequate duration of action, due to high clearance and a short half-life. Seen multiple times where drugs administered and has a half-life of 2 minutes. That's probably just not going to be long enough, at least in the real human world. As well as unanticipated drug interactions. A lot of times these are revealed in phase 2 and 3, and can result in very significant variability in the concentrations. So if you could, and by variability I mean really high, so Mike could take a dose of the drug, I could take the exact same dose of the drug at the exact same time, and due to just inherent differences in how we handle the drug, her concentrations could be 300 times higher than mine. And that's the type of variability, and I'll touch on this again a little bit at the end, is trying to assess that variability, quantify that variability, and if possible control that variability through pharmacokinetics. So between 1990 and 2000, if you look at this graph in the middle for pharmacokinetics and bioavailability, back in pre-91, approximately 40% of drug loss or attrition of drugs was due to poor pharmacokinetic properties. Now, due to enhanced screening methods and much better animal models, that has now dropped to around the 10% range more currently, or as of 2000. But efficacy still remains a very big, or lack of efficacy, a very big reason for attrition of drugs to this day. So PK and PD. So pharmacokinetics, PK, is the time course of the drug in humans. It's what the body does to the drug. Pharmacodynamics, PD, is the relationship between, I don't like to think of relationships with dose, I like to think of concentrations, but it can be the dose or the concentration of drug in the body, that relationship with the measured effect. So antihypertensive drug is taken, does it lower the blood pressure? If so, how much? And can you link the exposures, the drug concentrations or the dose, with that change in blood pressure that was measured? And so pharmacodynamics simply is what the drug does to the body. So the areas I'll be focusing a bit more on are focused on ADME, or the absorption, distribution, metabolism, and excretion. And these are just some boxes here showing these 4 different processes. All of them are critical in terms of assessing the pharmacokinetic parameters of the drug. So why are the pharmacokinetics important? This is a bit of a simplistic slide, but it really does illustrate why we need to do this kind of stuff. And so this is just an example of a drug that's given multiple doses. Each of these peaks is a different dose at 3 different doses. So here's a low dose, a middle dose, and a higher dose. The goal here, the ultimate aim of the therapy, is to achieve efficacy without toxicity. And so for each drug, even though it may not be defined, or the window may be wide, but each drug has a therapeutic index, a therapeutic window. What concentrations in the body are needed versus what concentrations in the body we need to avoid to prevent toxicities. And so, so this therapeutic window, or attempting to define what this window is critical for the drugs in different disease states. So absorption, obviously we think of that when we take a drug orally. I'm sure everybody in this room has probably taken a drug of some sort in the last month. Legal drug. I had ibuprofen a few days ago, so Claritin this morning because of the allergy things. But absorption is a very critical process in pharmacokinetics. It's simply the movement of drug drugs across the biological barriers from the site of administration to the bloodstream. Obviously, there are various routes of drug administration. Oral is probably the one we think of most of the time, but also parenteral, so intravenous, subcutaneous, and muscular. Other routes, inhalation, rectal, topical, transdermal delivery. And I don't know if you can read this. Go ahead. And bioavailability is also critical for a lot of the drugs. And that's simply the fraction of drug that reaches in the systemic circulation after administration from a route other than IV. Because if you give a drug intravenous, there's no absorption process, and boom, it's in the system immediately. So it's automatically 100% bioavailability for an IV drug. And that's when we look at relative bioavailability or absolute bioavailability to compare, for example, the area of the curve one would achieve from IV administration versus that area of the curve following oral or topical or some other route of administration. It does not dictate the rate of drug absorption, but it does dictate essentially how much of the dose is administered is going to get into the systemic circulation. So I've seen drugs, one of the early drugs for HIV had a 4% bioavailability. Awful, absolutely awful. It was heavily metabolized in the gut as well as undergoing first pass hepatic metabolism. And so the drug was sequinavir. And it didn't take too long to realize that another drug, I don't know if it came out first or second, it may have been second, ritonavir, was probably one of the most potent cytochrome P450, 384 inhibitors known to humankind. It's unbelievable what it can do. But recall, we have metabolizing enzymes in our gut as well. It's not just the liver. And so when this drug, sequinavir, was administered along with ritonavir, it increased the area under the curve by about 30 times. So that was what was administered, what patients took for years, was a sequinavir and ritonavir combination. And in fact, almost all of the subsequent protease inhibitor for treatment of HIV were subsequently had to be administered with low doses of ritonavir to block their metabolism, increase their half-lives, and increase their overall exposures. First pass metabolism is a critical step. As the blood supply of the upper GI tract passes through the liver before reaching the systemic circulation. And as I mentioned, drugs can be metabolized by the gut wall as well as in the liver. And so this first pass effect, if it's highly significant, it can really determine whether a drug is going to move forward or not. Hopefully this will be found out early on in preclinical, but at least in phase 1 to save a lot of money and cost and headaches later on trying to get something with such poor bioavailability approved. And again, just some of these slides are refreshers, I think, for some of you, but for acidic drugs, as the pH decreases, the amount of ionized drug decreases, and so the acidic drugs are better absorbed at the lower pH. Most of the drug in the stomach is unionized, favoring absorption. It's the unionized, the non-charged drug that's going to be absorbed. And for basic drugs, as the pH decreases, the amount of ionized drug increases. And so for basic drugs, they're better absorbed at higher pH. And most of the drug in the stomach is ionized. One of the other drugs that's approved for treatment of HIV had this very clear cutoff in the absorption of the drug. When the pH, trying to remember which way it went, but when the pH was less than 4, the absorption was perfectly fine. If it hit 4 in the gut or higher, so like taking antacids or something like that, the absorption was almost completely shut down. And so for that particular drug, it was important. One of the contraindicated drugs to go along with it would be H2 blockers or Malox, things like that, that would alter the gut pH. So those types of studies are critical as well. Distribution is simply reversible transfer of drug from one location to another within the body. The blood flows through the tissues and Equilibration is rapidly achieved with the heart, lungs, kidney, brain. Permeability of the drug in the tissues also affects distribution, so water solubility versus is a drug water soluble versus fat soluble. And drugs which pass, for example, the blood-brain barrier into the CNS are typically going to be small molecules and highly lipophilic drugs. So the physicochemical characteristics of the compound itself will dictate where it's going to go once it does get absorbed systemically. Protein binding is also a very critical factor for a lot of the drugs. They're transported as free or unbound drug and partly reversibly bound to various blood components. It's important to remember that only the free drug is the active drug. And two factors generally determine the degree of binding. Those are the affinity of drug for the plasma protein. So acidic drugs tend to like albumin, and basic drugs tend to like alpha-1 acylglycoprotein more in terms of protein binding. It also depends on the number of binding sites available. So drugs will compete with each other as well as hormones and other endogenous substances for protein binding sites. Many of the drugs that we could use, especially the protease inhibitors, the one I mentioned earlier was 99.9% protein binding. So that little itty-bitty amount of free drug was available and active to prevent HIV replication. So there's a wide range of degrees of protein binding for a lot of different drugs. Distribution volume, it's really a theoretical volume that would be required in the body to contain the administered dose if that dose was evenly distributed at the concentration measured in plasma. So we have to think of distribution volume because we measure everything in units per volume, milligram per liter, nanograms per mil, micrograms per mil. And so we have to have a volume. And just because a drug has a, for example, the same drug I was talking about actually has a distribution volume over 300 liters. And I don't know if we have 300 liters in this room in terms of volume. So it's a theoretical volume that has to be used, which accounts for the protein binding, tissue penetration differences, etc. So we generally think of drugs in terms of three different degrees, if you will, of distribution volume. Low, 3 to 5 liters, that distributes primarily in the plasma volume. So drugs with large molecular weights or strongly bounded proteins. Medium, 12 to 14-ish, distribute in extracellular space. And then high distribution volume drugs, typically thought of as greater than 40, 42 liters, distribute in tissues. And drugs stored in fat may have distribution volumes much greater than total body fluid. Thus, the example of a drug with 300 liters, it doesn't sound possible, but it just describes where the drug is going in the body. Elimination, again, we can think of this primarily in terms of metabolism and excretion, various routes of excretion of the drug, but it's an irreversible loss of the drug from the site of measurement. Metabolism, the conversion of one chemical species to another, and excretion, again, the irreversible loss of chemically unchanged drug. This graph over here simply shows, especially on this side, a lot of the different isozymes, cytochrome isozymes that are used for drug metabolism, liver uses for drug metabolism. Clearly, CYP3A is the most predominant of these isoforms that handle drug metabolism, particularly through the liver. So enzymes involved in metabolism are present in many tissues, produces active or inactive metabolites. Overall goal. So for example, if any of you had the unfortunate pleasure of having the flu this year or this season, Tamiflu is oseltamivir. It's actually a prodrug. So oseltamivir phosphate is the parent, that's the part you swallow. But once it gets into your system, it's actually converted into oseltamivir carboxylate. It's the primary metabolite that's active against influenza replication. And so understanding those processes is obviously critical, especially if a prodrug type of compound is administered and one of the metabolites is the active component. And drug metabolism rates vary greatly among people. There's genetic factors, obviously, that can cause this, disease, coexisting diseases, and drug-drug interactions can drastically change the systemic exposure of a drug. So again, just a little bit more background on the types of metabolism. Again, they're broken down into phase 1 and phase 2 metabolism. Phase 1 involves formation of new or modified functional groups through oxidation, reduction, or hydrolysis. This is the cytochrome P450, primarily cytochrome P450 system. It's a microsomal superfamily that catalyzes the oxidation of many of the drugs. There are multiple different families within this group, and the enzymes can be induced or inhibited by many drugs and other substances. So when you have a compound that is exclusively or primarily metabolized by one of the primary isoforms, understanding drug-drug interactions is going to be critical at some stage because interactions very, very likely will occur given all the other drugs that are available. Again, most drugs are metabolized by P450-384 isozymes. Some drugs will induce or speed up P450 metabolism, which will result in a decrease in the plasma concentration of the other drugs. So this is induction, and some drugs will inhibit or slow down the P450 metabolism and thereby increase the plasma concentrations of the other drugs. That's what this ritonavir drug I mentioned does. It almost just shuts down P450-384. So anything going through that pathway, it's not getting metabolized very well at all, and the plasma concentration exposure significantly go up.
Do drugs do that by modifying the enzymatic activity or by regulating the expression of the enzyme, just blocking the enzymatic?
Primarily. Phase 2 reactions then occur because many of the phase 1 metabolites are too lipophilic to be retained in the tubules. So conjugation reactions with an endogenous substrate or amino acid results in more water-soluble compounds. So these include glucuronidation, sulfation, and acetylation. And again, the whole reason for metabolism of xenobiotics is simply to create a more polar compound that can then be excreted and eliminated from the body. And that's what these phase 1 and phase 2 reactions do. Excretion is the elimination of unchanged drug from the body. Drugs may be eliminated. We typically would think of the kidneys, renal elimination, the primary source of drug elimination, but other routes are available such as lungs, saliva, sweat, breast milk. And also enterohepatic circulation can and does occur with some drugs. So they can be excreted in the bile, stored and released from the gallbladder, transit into the small intestine, and then reabsorbed into the circulation. And you can see this graphically if you're doing a study, an intensive, what we call an intensive collection of samples following drug administration. The concentration time curve goes up, starts coming down like it's supposed to, and then it goes, whoop, comes back up again, and then goes back down. So it creates this double peak phenomenon in the concentration time curve. And when you see it, you pretty much know it. Yeah, it's there. So renal excretion, several components: glomerular filtration, tubular reabsorption, and tubular secretion. And these are located, described over here with these different with the numbers 1 through 4. And so glomerular filtration. I'm just going to skip to the next one. So why this is important is the renal clearance can be calculated to investigate the mechanism of drug excretion. So if the renal clearance is approximately 120 milliliters per minute, then the drug is filtered, glomerular filtration, but there's no secretion going on. If it's less than 120 mils per minute, there's filtration, glomerular filtration occurring, of course, but also tubular reabsorption is occurring. If the calculated renal clearance is greater than 120, then filtration and secretion are going on at the same time. And so we can break down clearly to do this. urinary pharmacokinetic studies generally are going to be rooted. Sometimes we can do them and sometimes we can't. It seems easy because it's not like drawing the blood, pretty much pee in a cup and you're good to go. But it does require somebody to hang around for a while, at least from a patient's perspective. And a lot of times that's not too much fun when they can just go in and have a blood stick and leave. And I would venture to guess in some animal models it may not be too easy either. But total clearance and half-life. Clearance is really, it's measured the ability of the body to eliminate the drug. It's not an indicator of how much drug is removed, but it's the volume of plasma cleared of drug in a given period of time. So it's always going to be expressed as volume per unit time, mils per minute, liters per hour, are generally how we report clearance. It's probably one of the most important parameters, pharmacokinetics speaking, that we have to take a look at. If we do, for example, drug interaction studies, is the clearance different between the drug itself and the drug given with another drug that might be interacting with it, as well as the area of the curve and other parameters. Half-life is also very important. For a lot of the animal work that I've been involved in, people that I've worked with, they have this tunnel vision on half-life. I can understand that. Again, if it's 30 seconds, it's probably not good. If it's 10 hours, then a little bit better. But the half-life is simply the time for plasma concentrations to be reduced by 50%. And generally, 5 half-lives, after starting a dose, 5 half-lives is what it would take to achieve steady state, as well as once the drug is stopped, 5 half-lives is what it takes for the drug to be completely removed from the system. And so those are rough, good but rough guidelines for thinking about the half-life. And the half-life also is going to, as I'm sure you know, be influenced greatly what the dosing interval of a drug might be. So a really good example of this is in the early days of HIV therapeutics with AZT, zidovudine. The drug itself had a very rapid half-life. And because of that, it was actually dosed 5 times a day. Then people realized, well, it's not the zidovudine parent drug that's active, it's the intracellular triphosphate form of the compound that's actually inhibiting viral replication. So they started to look at that and realized that the half-life of the intracellular triphosphate is actually much, much longer. And so they ended up going to 3 times a day dosing to ultimately have to twice a day dosing with the right dose and the long half-life of the intracellular component. So the half-life will play a major role in what the dosing interval of a drug will be. In a lot of the areas I work in, it's either once a day dosing or the drug doesn't move forward. Twice a day, maybe. But anything more than that, the drug will die in phase 1 or 2. Just because patients aren't going to want. I mean, think about it. Can you really take a drug on schedule 3 times a day for 10 days or longer for the rest of your life? Very difficult to do. So total clearance, again, it's just a combination of the renal component, the hepatic component, and any other clearance components. And enzymes and transporters are subject to inhibition and induction, as well as genetic polymorphisms that can alter the clearance on the drug. So we have some examples in antiretroviral therapeutics where genetic polymorphisms in the metabolizing enzymes clearly affect the clearance of the drug across different populations who may have more, express more of that genetic polymorphism. So that, and that's really a lot where the whole pharmacogenetic component comes in, at least from my perspective anyway, in terms of drug metabolism and clearance. So a lot of this, its application to drug development. So I wanted to include this for both preclinical and clinical studies. I once saw a professor present the seminar. I couldn't tell you what it was about at this point. where he had, I think it was with mice or rats, was looking at some effect of a drug. He used 0 milligrams, so placebo, 20 milligrams, and 40 milligrams, and tried to convince me that that was a dose-ranging trial. That's twofold. That's it. That is not a dose-ranging trial. You might as well just look at one. And so, if anything that you might get out of this, understand that a dose-ranging trial has to be a dose-ranging trial. Whether it's in animals or cell lines or humans, a wide range of doses needs to be looked at, or you will never, ever, ever find concentration or dose-response relationships. And so for these studies, again, animals or humans, the single ascending dose and multiple ascending dose type study designs are needed using an adequate dosing range. This is to understand what the kinetics look like. Again, if the drug has severely nonlinear pharmacokinetics and there are, QTC interval changes or some cardiovascular potential or other toxic potential for the drug, that drug is basically over. There's really no reason, there's not much of a reason to continue looking at a compound like that. But a very bare minimum of 3 doses over a greater than or equal to, I would prefer much greater than a 10-fold range of doses is what needs to be looked at. Obviously, using an adequate sample size, again, it doesn't have to be huge. 6, 8 mice. Study I'm going to show you, I'm going to wrap up here with, was 8 patients per dose group for 3 dose groups. Perfect study. They just nailed it. But collecting samples over an appropriate time frame, and then most importantly is examining the concentration-response relationship, as opposed to, I'm not going to try to pick on anything here, but I still can't quite grasp why in cancer therapeutics a drug in the drug development process is dosed until maximum tolerability, and then they bring it back a dose and say, here's a dose. Now, if we don't have a biomarker, I can understand that, but in some cases now, especially over the last 5 to 10 years, various biomarkers have really become much more prevalent. And so why not look at a wide range of doses? If it's a solid tumor, maybe it's a certain shrinkage in tumor size. There's a whole different ways to quantitate the effects of the drug, as opposed to just dosing it until everybody gets really sick, and then coming back a dose and saying, voila, here's our dose. So myself, and I know there's plenty of others in the pharmacometrics world that would like to see more concentration and or dose response type examinations of a lot of these drugs. It really helps to very clearly identify what the dose is and the dosing interval for that drug. So how are the pharmacokinetics of the drug determined? Again, I don't want to bore you too much with some of this, but I'm going to go through it anyway. First, you have to think about the sample collection. Yes?
So you talked about different doses of the drugs, say in animals. So can't we choose the cell cultures to do the different doses and then come to the dose and then give that particular dose to the animal?
Well, do the cell cultures handle the drug the exact same way as the animal? So I don't know, yours might, I don't know. But that's the difference, is even if you're giving the drug intraperitoneal or however you might be giving the drug to the animal, if it's not IV, you're going to have an absorption issue. In cells, well, you guys can answer better than me, you probably don't have as much of an absorption issue. Maybe some permeability issues, but my opinion is that's not the best way to do it. I think you can do that in cells to examine from a big picture perspective, what is the lowest amount of drug needed to elicit some type of whatever the effect is that you measure, versus the other end, what is such a high exposure that all your cells die, whatever the toxicity might be. And then from within that range is where you go from to the animals, and design a wide dose ranging study.
So a minimum of at least 3 doses in animals.
You have to be careful. I'll touch on this a little bit more at the end. A lot of this is artwork as much as science, and so with a lot of experience, I think 3 doses is reasonable. If you've done this kind of stuff a lot before, if not, I still personally like 4 doses. Because you have a much better, and again, I'll show this a little bit, you have a much better opportunity of capturing the concentration response, the full concentration response range or curve, if you will. And so, but the key really is the range. 0, 20, and 40 milligrams. It's 2 times, and it's not good. Now, if he had done 2, 20, and 40, that's a 20-fold range, 20 times. That's much more reasonable. That's actually the range I like to see. It's closer to 20 times difference between the lowest and the highest dose, with at least 1 in the middle. And oftentimes, at least in clinical trials, this is where the FDA would get involved. We're dealing with some of that right now, where they would say, I want to look at low dose, middle dose, and high dose in phase 2A. But that middle dose is critical. They generally like it to be a little bit more closer to the lower dose as opposed to closer to the higher dose, just more accurately get that concentration response relationship.
So final question. Sure. Let me extend to the question. So how well can you this PK, PD data from animal study predict PK, PD in human patients?
That's a great question. For some drugs, very well, for others, not well at all. And we can do the various modeling strategies, techniques, but in the end, it still comes down to, and this happens going from adult patients down to children or babies, how you make that transition. A lot of times what we do is make the best guess that we can, try it in a very small number of people, see what it looks like. If we're way off, we say, well, okay, well, now we know, and then alter your doses. So every one of the pediatric protocols that I'm involved in, that's how it's set up. So if we think this is roughly the right dose we need, but we don't know. So we're going to look at this dose first, but allow room in the protocol to change it if the results don't fit what they should look like. But yeah, that's a great question. So but scaling from animals to humans, there's software out there, there's various ways of doing it. But
it also depends on which animal you use. Right?
Correct.
Primates probably
yes.
do better.
Primates, so in general, what I've seen in my area, primates have been better. But it's still, again, and I'll touch on this a little bit at the end, I'm sure you won't want to see, but that's where the modeling work really comes into handy. It comes into play. It's making some assumptions and then going from an animal model to humans of what it might look like. So anyway, sample collection, generally what we do is it's going to be invasive. We need blood samples. We measure drugs and plasma, but clearly other samples can be collected. We have to think of intense versus sparse sampling. So intensive sampling is going to be where, again, from a patient perspective, a patient will come into the clinic or in the research center, and they're there for 12 or 24 hours, and you're collecting 10, 15 samples over that time period. That's an intensive collection. Sparse is going to be where that same patient would come in, but might only get 1 or 2 samples drawn, and then they get to leave. But they have to come back more frequently for those 1 or 2 samples. So, and sparse sampling is generally designed to be used for population pharmacokinetic analyses, where we don't have as many samples on each person, but we have a lot more people. We can put it all together in a model, as opposed to intensive, where we have much fewer people, but we have a lot of time points for each one. And so that's really some of the thinking in terms of the sample collection for study design. Obviously, there's other non-invasive methods, and most of our techniques are designed for plasma, although we've done all of those other ones except for feces, I think. We've we're now doing quantitative drug from various tissue biopsies, a lot of different areas. You have to think of the analytical methods. This is really what my lab does. We have both HPLC as well as primarily mass spectrometers in the lab. HPLC is fine. I generally think of this, or UPLC, as they're good methods. They're not going to be as sensitive. So a lot of times, especially if you're dealing with animal, like mice, you can't obviously suck a lot of blood out of a mouse. You can get some. So you're dealing with tiny sample volumes. Same with some of the pediatric studies that we do. We can't get, and you'd be lucky if you get 0.2 milliliters of blood from one sample. And so a lot of times, depending on the method, the HPLC is not going to be sensitive enough using such small sample volumes. And so our field, anyway, has moved much more over to mass specs. The problem with them is they're outrageously expensive. You know, if anybody has an extra 400 grand laying around they don't need, come talk to me. They can be difficult to run and expensive to maintain, so you need personnel in your lab who are really experts at running mass specs. And I've been very fortunate to have a crew of them for many years. So this is a representative pharmacokinetic profile. This would be from an intensive sampling setup, for example, where each triangle is where a blood concentration is drawn for this drug over a dosing interval of 24 hours, so once a day dosing. But the peak concentration is reached for this drug around 5 or so hours, and then the drug is eliminated over time. This particular drug has a half-life of roughly 10 hours. But the absorption phase, obviously over here, the elimination phase, this is where you calculate the half-life from. And then in our field, anyway, the trough concentration, so the concentration measured at the end of the dosing interval, is very critical for maintaining therapeutic efficacy and preventing the development of resistant isolates. And so that would be the C24 or the C-min at 24 hours. And then this whole area under the curve is the AUC, the area of the curve for 24 hours. And so once you have these parameters, this would be what this slide is showing is what we call a non-compartmental pharmacokinetic analysis. So we have each of the time points. In this example, it's just a dosing interval is 8 hours. But we have each of the time points, and we simply apply the trapezoidal rule to calculate the area of the curve. And so it's extremely simple for a non-compartmental analysis. So we're not applying any models to the data yet. It's just a simple, straightforward non-compartmental way of applying the trapezoidal rule to calculate the area of the curve. Once you have the area of the curve, there are equations everywhere to calculate all the other parameters that you could possibly need. So you know the dose of the drug, you know the AUC, you can calculate the clearance, you can calculate the elimination rate constant from the tail end of the curve. From that, you can get the half-life as well as distribution volume and all the other parameters that one would need to describe the characteristics of that drug. Clearly, I don't sit there and do all these every day. We have computer programs that make it really, really simple. But we need to know the basics behind what the computer programs are actually doing. This then leads me into modeling. And again, this is for humans as well as animals, and I think we've done some, from various cell lines as well. But it's simply just applying a set of differential equations to describe the concentration time curve of the drug. And it has some, and that's our type of modeling in its most basic form. But it has some very, very important aspects to modeling that you might not always think of, that I just want to make sure that you're made aware of. But some of the philosophies are to describe the data, quantify processes, explore mechanisms, and most importantly to me anyways, make predictions. So we can get the area of the curve, clearance, volume, half-life, all that stuff from a non-compartmental analysis. We get all these from modeling also, but when we have a model, we can now also start to assess covariance. age, weight, renal function, hepatic function, whatever you might want to include in there. And these covariates are there to help describe the variability. And we can describe absorption characteristics. A lot of times you see for drugs, I don't have one off the top of my head, but you look at the labels for a drug and it'll say take with food, or take on an empty stomach, or, you know, don't take with a double cheeseburger, you know, something like that. And the reason is the absorption phase characteristics of the drug. And so modeling really helps describe, you know, is that absorption phase super fast and going up, or is it now much slower, and assessing and affecting the overall drug exposure. Explore metabolite formation rate constant. So if you have a parent drug or prodrug that's metabolized to an active metabolite, what is that process in between? So the modeling will help us do that. We saw for also Tamiflu in babies, where the conversion from the parent, oxytamivir phosphate, to the carboxylate, there were two distinct populations in a group of about roughly 80 0 to 2-year-old babies. And one had they just went straight from the parent to the metabolite. The other group had this roughly an hour lag phase before the metabolite was actually formed. And by modeling and looking at the difference, I finally concluded the reason that was occurring was these babies, it was occurring more often in the smaller babies, closer to 0 to 6 months, years of age. Well, what are smaller babies doing? They're getting fed much more frequently than older kids. And that's exactly what it was. Is the feeding was more consistent or more frequent in the younger babies. It was actually affecting the rate of conversion to the active metabolite. Now, the overall amount of drug there was similar between the two groups, but it was very interesting, and that led us then to include age as a covariate when we were modeling the data. But importantly is also simulating the concentrations to predict effects and directly linking the pharmacokinetic model with whatever the response is. And our field really has gone, has come a long way, especially in terms of this last point. I didn't really include any of the slides here, but linking the PK with the response and conducting simulations is really catching on, and luckily, and I think it's going to be an important way to move forward. But so just, again, very basic, when we're thinking of models, this would be a one-compartment model. So the drug is administered. Here we've got this, like an IV injection into this little thing. Before administration, and almost immediately after administration, the drug is homogeneously distributed throughout the entire body. And graphically, from a differential equation type perspective, this is simply what the model would look like. Graphically, this is what it would look like. So from the peak concentration right around here until the last measured concentration down here is essentially a straight line. So there's no distribution phase. It's just all the drug is almost instantaneously throughout the entire body. As opposed to a two-compartment model where the drug is administered and then goes to various sites in the body first. Again, it could be the heart, brain, lungs, it could be fatty tissue, it could be bone, it just depends on the drug. And then it's distributed to the rest of the body. Graphically, this is what a two-compartment would look like. Right after the peak, there's a very sharp drop. This is the distribution phase in the concentrations. And then it levels out after that. And that's the terminal elimination phase of the drug. So if you see a curve with this, that's not a straight line from the peak to the last concentration, it may very well be a two-compartment drug. And we can have 3 and 4, we can have there's a whole, nothing saying the drug has to be 1 or 2. It just depends on the drug. Again, I'm not going to spend too much time on this, but this is the Tamiflu example that I was talking about. So what we did, now that you know what a 1 and 2 compartment model look like, this is the parent drug, oseltamivir phosphate, that we modeled using a 2-compartment disposition. and then a delay phase to the formation of the metabolite, and then a simple one-compartment model for the description of the metabolite. We assessed the model fits. This is all a population pharmacokinetic, mostly design, but we look at this in terms of goodness of fit plots, measured versus predicted, typical type of way, as well as visual predictive checks where we will simulate, we have the model parameters, and we'll simulate 500 or 1,000 different concentration time curves, and then compare it to the actual data that was measured, and make sure that they stay within a certain parameters to ensure that we have a good model fit. But this is then where we would also, where we look at the covariance to see how these curves down at the bottom would be affected by age, weight, and other components. So lastly, I just want to end up with what I think is one of the best examples of a phase 2A proof of concept study, at least within the realm of antiviral drugs, that I've seen. This is a drug called dolutegravir, relatively recently approved by the FDA for treatment of HIV infection. They conducted this proof of concept study looking at 2 milligrams, 10 milligrams, and 50 milligrams. 3 dose groups, 8 patients per dose group, as well as a placebo group. This was monotherapy. This is the only drug that they received, and it was administered for 10 days, once a day for 10 days. And they quantitated then this, the change in viral load in plasma HIV RNA daily, at least, over this 10-day period of time and beyond. And you can see a very nice differential between all 3 of these doses. And that's what you want to see, whether you use 3 doses or 4 doses or something else. If you don't have a wide enough dose range, all of these lines are going to be really close to each other, and you'll never find the concentration-response relationship. Every single drug has one. It's just a matter of finding it. And so that's why. So if you think about this, this is a 25 times the dose range, right? Going from 2 up to 50. Nice, wide, beautiful. They pulled it off with 3 doses, which is pretty amazing. But the reason this is important to have these separations in the response is to analyze the concentration response data afterward. So here is just the concentration time curve, the pharmacokinetics of the drug at those 3 different doses. Very simple, one compartment looking drug. This over here is the trough. So this is the C24, the concentration measured at the end of the dosing interval, versus the change in the viral load from baseline to the end of day 10. And this is what they're able to find. So this group down here is the 2 milligrams, these blues are the 10 milligrams, and these greenish ones are the 50 milligrams. They were able to fit a beautiful Emax curve, maximum effect curve, through this data set. And why is that important? Well, you not only determine what the maximum effect of the drug is, which is the flat part,